Neuropharmacology. This is a video made by me as a summary of the Sci 354 course from Macquarie Uni, the neuroscience course. What is neuropharmacology? It is the use of pharmacology, which is the study of drug interaction on receptors, to assess the functional neurons and nervous system. We must consider receptor activation, whereby drugs that alter neurons directly can include receptor agonists that bind to receptors to cause an effect, and receptor antagonists that bind to receptors but it doesn't have an effect, and it just merely stops the neurotransmitter from having an effect as it blocks the way. Drugs that alter neurons indirectly are called positive allosteric modulators, or PAM. These enhance the function of agonists at the receptor. Negative allosteric modulators, or NAM, reduces the function of an agonist at receptors. Hormone release. There are two primary glands or areas in the brain that are important for hormone release. These include the pineal and pituitary. The pineal gland releases melatonin, while the pituitary, which is regulated by the hypothalamus, releases subsequent hormones. The anterior pituitary. This is controlled by the parvocellular secretory cells of the hypothalamus. The anterior pituitary releases adrenocorticotropic hormone, or ACTH, that is regulated by the hypothalamus, which releases CRH. As a result of this release of ACTH, the adrenal cortex controls cortical release, and this can suppress immune system and mobilize energy storage. So in the case of cortisol, it is a stress hormone, and it is used to indicate how much stress there is in the body. Other hormones of considerable note can include the follicle stimulating hormone, or FSH, which is produced in the gonads, and these influence ovulation and spermatogenesis, gonads being the sex organs. Luteinizing hormone, or LH, are gonads that influence ovulation, or testosterone. Thyroid stimulating hormone, or TH, is secreted by the thyroid thoraxin to increase metabolism. There is also the growth hormone, or GH, which affects all cells and stimulates protein synthesis. Finally, there is prolactin, which influences mammary glands, allowing for their growth and subsequent milk secretion. Oxytocin. This is a neuropeptide hormone that is produced by the magnocellular neurosecretory cells in the hypothalamus. It is released into the bloodstream of the posterior pituitary gland. Oxytocin in the periphery. Oxytocin controls myometrical contractions in childbirth. It increases sperm number and contractions in ejaculation, and it contracts myoepithelial cells to form and create breast milk. Oxytocin in the brain. It is produced in the magnocellular secretory cells in the hypothalamus, which I mentioned prior, and from there it reaches the paraventricular nucleus, or PVN, which moves via the neuronal contact. It also reaches the supraaortic nucleus, or SUN, that moves via volume transmission, which aids in the somatic dendritic release of oxytocin, and it also affects the accessory nucleus. Oxytocin and addiction. Kolb in 2008 found that oxytocin via MDMA consumption served as an anxiolytic, anxiety reducer. Oxytocin injected into the subthalamic nucleus, or the nucleus accumbens, was found to reduce meth reward, hence reducing the effects of addiction. Vassopressin, aka antidiuretic hormone, ADH. In the periphery, ADH retains fluid in the body via V2 receptors. It regulates blood volume and salt concentration, controlling for constriction of blood vessels, and increases blood pressure in V1 receptors. The effects of vasopressin in the brain. There are high numbers of vasopressin receptor sites in the red nucleus, central amygdala, and nucleus accumbens. This was found by Cobb in 2008. It was discovered that activating V1A receptors produced prosocial effects in animals, while activating V1B receptors produced more anxiety, fear, and aggression. V1A receptor antagonists reduce the effect of oxytocin oxytocin, increasing the chance of meth relapse. So hence, blocking V1A receptors was able to produce those symptoms of addiction to drugs and relapse back into the addiction state. Moving over, we should also look at molecular neuropharmacology. Is this the new meta? Basically, molecular neuropharmacology is the understanding of neurotransmitters or drugs and its effects at the molecular level. There are new techniques that have been used to learn more about neuropharmacology. These include fast scan cyclic voltammetry or FSCV. These measure electroactive neurotransmitters like serotonin, dopamine, and noradrenaline, or any other chemical that undergoes oxidation. They use carbon fiber microelectrodes that are inserted into the brain region of interest, and a voltage is applied. The voltage is added, and there can be positive oxidation or negative reduction. As a result of the voltage data, a waveform is triangulated at 100 ms, and the advantages of FSCV is that each compound has its own voltage level and there is good spatial/temporal resolution. ACH and addiction. ACH cells are located in the magnocellular basal forebrain system and brainstem system. They project around the brain using volume transmission, and ACH is involved in reward, addiction, memory, learning, muscle movement, and REM sleep. The injection of ACH and glutamate receptor antagonists can block the effects of cocaine, and ACH also regulates the release of dopamine in the VTA, the ventrotegmental area. And yeah, this is neuropharmacology. So in summary, we looked at what is neuropharmacology, how hormones are released, receptor activation, oxytocin, vasopressin, molecular neuropharmacology, and fast scan cyclic voltammetry as a technique used, as well as ACH and addiction. So thanks for watching.